Hi everyone, this video will cover our second lab report, the Drosophila Population study. So this is our one of our long-term studies that we've been working on throughout the course. So let's take a look at what we are studying. So just like the first lab report that we did on the wavelengths of light, we're going to do the same format. So our lab report needs to be, the same structure with a title, with an abstract, with an introduction section, a method section, results discussion, conclusions, and then finally the reference section. So we need to have each one of these sections present, just like our first lab report. So as far as the title, we actually gave it to you. So luckily, you have it there. So it says in your lab manual, it says the title should be Drosophila Population Study. So that will be your given title. So you can just, write that or type it at the top of your page. So that's pretty easy. It's already there. Next, you have to have your abstract. And just like with the first lab report, I highly suggest that you complete this after you've done the other sections. Because again, it's kind of like a summary of your lab report. So you're summarizing everything that you included in your lab report. So what you did, how you did it, an overview of the results, and your conclusion. So since we are basically taking a look at this experiment where we looked at how the amount of space, the large container versus the small container, and the amount of food, the large amount of food versus the small amount of food, affect the growth of a fruit fly population. So, how did we set it up? And then kind of give a brief overview of the results and your conclusions. So just a paragraph of the experiment itself. Okay. Next, we're going to get into the introduction. Now, let me go into our Moodle page, because in our Moodle page, if we go down into our topic 7, so you'll see here, okay, so the Drosophila fruit fly population link, this is where you're going to be submitting your lab report. But right above it, it says concepts to include. There's a list of different concepts to include. So I've kind of summarized some of these for the introduction. Some of them could go in the method section. Some of them can go in the discussion section. So, keep this handy, and you might want to, define some of these key terms throughout your paper. Okay. So you might want to print it out or something like that and have that handy with you. Along with in your lab report, or in your workbook, on page 291 is where there's basically the information for your lab report. It's detailed information of what to include. So let's go back. So let's start with the introduction. So the beginning of your introduction, you're going to establish the research problem. So what question are you trying to ask? So in our workbook on page 291, it gives us the question. It says, how does space and amount of food affect the growth of a fruit fly population? So that is our question. So you'll need to include that question as part of our introduction. Okay. Then we need to establish scientific concepts of the lab. So what background information should the reader know? So here is some information that the reader should know. So we need to describe the fruit fly life cycle. Since we are taking a look at the growth of a fruit fly population, we need to see what the fruit, how long is the fruit fly life cycle? How long does it take for the fruit flies to go from mating to laying eggs to, growing into a full-fledged adult fruit fly, to start the cycle over again. So you'll need to, describe that fruit fruit fly life cycle. It's also important to talk about what is population growth. What are some environmental effects of population growth? What limits population growth? So this is where we get into the concept of carrying capacity. So think of those density dependent factors and the density independent factors that can limit population growth, prevent it from going over its carrying capacity, and that relationship between the environment and the carrying capacity. You'll also want to talk about those growth curves. So, what type of growth exhibits a J-shaped curve versus an S-shaped curve versus the boom and crash cycle. So, think you might need to describe those types of population growth rates. And then finally, you'll probably want to discuss the life history patterns. So what are R selected species versus K selected species, and what are their reproductive strategies? So all of those different concepts, we need to lay that foundation down for the reader, because that will apply to what we know about the fruit fly, and it'll help us understand what we see in the results of the experiment. Now, we need to include a hypothesis. So, towards the end of our introduction, we'll need to state our hypothesis and provide logical reasoning. So, basically for this, the question again was, how does space and amount of food affect the growth of a fruit fly population? So essentially what we're asking or answering is, which container combination will have the largest population after the 6 weeks? Okay, so remember, this was a long-term project, and over each week we gave you the fruit fly populations for each of the containers. And so we had 4 containers. One of the containers was a large container that had large amount of food at the beginning. And then we had a large container with a small amount of food. And then we had a small container with a large amount of food. And then we had a small container with a small amount of food. So instead of coming up with a hypothesis now, you'll want to refer to your original hypothesis at the beginning of the term. So you'll want to go back to topic 2, step 8 in the forums. So we'll want to go back into Moodle and go back to topic 2, step 8. Oops. It always kicks me out. Okay, so this is where this is early in the term, so nobody's posted anything, but where we have posted all of our hypotheses. So take a look at what your original hypothesis was, okay? So which container do you think was going to show the largest population increase, and why? Okay, so that will, kind of refer to that. So that will be your hypothesis. Okay, so no cheating, no, looking at what the final results were. We want to kind of go with your original reasoning. Okay, now this is different from our first lab report. We're actually going to do another hypothesis. So based on our original hypothesis, we're also going to include a graphical hypothesis. So again, if you refer to page 291 in your workbook, it will talk about how to construct this graphical hypothesis. So it says to use graph paper to draw a population curve that represents your written hypothesis. So in this case, it's okay to hand draw it and submit it as a drawn thing. And I don't mind if you want to do a digital copy, but it's going to be a lot easier to hand draw it because this is going to be your hypothesis. So digital one, it's going to be a lot harder to draw because you're going to have to come up with random numbers. So that's why hand-drawn one is best. So on page 293 is a piece of graph paper, and you're going to set it up where you're going to have all 4 curves will be on the same graph. So you'll have different colors representing each of the populations. So if I go back one slide, so you'll have one color that will represent the large container with the large food, a different color representing the large container with the small food, and so forth. Okay, so 4 different colors representing the 4 treatments. On the x-axis will be time, so we'll have the 6 weeks that we measured, and then on the y-axis will be the number of fruit flies. So use your hypothesis from the previous, topic 2, step 8, and draw, what you think your hypothesis, draw it out as what you think your hypothesis represented. And then do that with the other populations as well. And then explain why you think each curve will appear as it was graphed. So all of this is part of the introduction. So it's kind of strange that you're going to have a graph in your introduction, but that's just what was required for this particular lab report. All right. So that's the entire introduction with a graph. Next, you're going to have your methods section. So the methods section, again, we didn't perform the experiment, so we're just kind of going off of what is written in the manual, in the workbook. So you're going to go through pages 295, 296, and 297 to kind of see how the experiment was set up. So mainly, we want you to kind of look at how the containers were established. So you want a written description of like the physical environment, so the space part of it, and the nutritional environment. So what how did they do the food part of it, and how did they do the space part of it? So what was the difference between the large and the small container, and then what was the difference between the large amount of food versus the small amount of food? And then, so we got to kind of establish the treatment, the independent variables there, and see, what was the difference between them and so forth. And then don't forget to cite the workbook, since the workbook is what is supplying that information. And then finally, we're going to look at the results. So the results were given to you, and let me go back. So if we go back to our Moodle page, and where did I give the results? Nope, I think I gave it in this one. Yeah, so here was step count for step 4, and then here was the final one. Okay, so the final one was was given in week 6, and so you'll have all 6 filled up. And then you're going to be graphing them. So once you take all of this information, then you're going to convert it to a graph. So again, you can draw it out, hand draw it, because it says so in the lab manual that it's okay to go ahead and hand draw it. So it'll be a single graph, just like you had for the hypothesis, okay? So the graphical hypothesis will be the same setup as the results. So you'll have on the x-axis will be time, so it'll be divided by the weeks, and then the y-axis will be the number of Drosophila. And then I recommend using the same colors for the same treatment groups, okay? And then draw the curves. So that will be the graph, but I got ahead of myself. So before But for the results section, you always open with effective overall findings. So what is some general trends that you saw? So one thing that you might want to state is, did all of the populations survive? Did one stand out as a clear, one that ended up surviving better than the others? So you don't have to, go into detail, but you can say, that this treatment group seemed to outlast the other ones by far or whatever. And then you'll do your graph. So this will be very similar to the graph or the setup for the graph that you did for your graphical hypothesis. So you'll have all 4 lines on one graph, include a title, you'll need your axes label, and your legend present indicating what color goes with what treatment group, what container. Okay. So hand-drawn is just fine, just like you did for the graphical hypothesis. And then in addition to your actual graph, you'll need to have a written finding paragraph that clearly talks about what the graph is representing. So describe what trends and data do you see? Okay, so this is an opportunity to, talk about, what type of growth curve you're noticing for each of the curves. I mean, are they J-shaped curves? Are they S-shaped curves? Are they boom crash cycles? What type of population curves are you noticing? Do you see a trend? Is there, one that is standing out? Which container seems to, did a population end up dying off? Do you think just what are the trends that you see? Okay. And then finally, don't forget to label your graph. So this would be a graph. So that would be graph 1 or figure 1. And then refer to that in your written findings. And then if you want, you can type up the raw data, the chart. So if you're going to do the table, you need to label it as a table, and then make sure that you include the table as table 1 and then refer to that in your findings as well. And then don't forget to include figure captions and table captions for your figures and tables, and refer to them within the text. And then finally we get to the discussion. So we always open our discussion with a reminder of what our original hypothesis was for the, which container was going to end up having the largest population in the end, and then deciding if the data supported our hypothesis. So which ended up being the one that had the highest population. Does that support our hypothesis or not? And then we need to back up our statement with reference to appropriate findings. So this is where we get into some of those keywords or key concepts from our Moodle page. So I kind of, brought in some of these questions that you can ask yourself. So how did the environmental resistant factors affect the populations? So, think about the space. Do you think a larger container affected the populations differently than a small container? Why would a larger container maybe affect the population in a way differently than a smaller container? How did it affect it? The large amount of food versus the small amount of food, how would that have affected the populations differently? Were there any unexpected results? So if you look at the 4 different treatment groups, Was there one that you completely thought was not going to work and it worked? Or the opposite, maybe one that you thought was going to be the leader and did not, and suffered completely? What was there anything that was unexpected to you, and why was it unexpected to you? So as part of the discussion, we also need to kind of link the answers to the research question to solutions of a problem, as well as information for the field and other applications. So what that means for our particular lab report would be answering these particular questions. So what type of growth curve did you observe and why? So was it, that J-shaped curve, the S-shape, or boom and crash? And then looking at those growth curves, what was the rate of growth? Did you see it, go up steeply, or was it more of like a kind of a stable growth? And then did it, sharply, population decline sharply, or was it kind of a gradual decline and so forth? And then, was that the same for all 4 containers, or was it different? Did one container kind of slowly grow and the others just shot off? That kind of thing. So you want to kind of compare the rates of growth and decline for all 4 containers. And then you'll also want to bring it back to the survivorship curves. So, which survivorship curve applies to our containers? Was it, type 1, type 2, or type 3 survivorship curve? And relate that to the reproductive strategy of the fruit fly. So in the introduction, you talked about the life cycle and the, difference between an R strategy versus a K strategist species. So based on that information, what type of survivorship curve did the fruit fly have, and how does it relate to its reproductive strategy? And then also think about, with the curves that we created, do you think the fruit flies ever reach their carrying capacity? So explain why or why not. So, think about those density dependent and density independent factors that came into play, and think about, did they ever reach their carrying capacity? And then, tie that into the human population. So what correlation can you draw to our own worldwide human population growth? Can we, do you think the same thing will happen to us? Do you think our population will grow to a certain point and then crash? Have there been human civilizations in the past that have exhibited similar patterns as the fruit fly populations in the containers? So you can kind of relate these or possibly relate them, find a correlation to human population growth. And then finally, we need to sufficiently address other issues pertinent to the lab. So I thought of some. So what were some sources of error in this experiment that you could think of? So think about like the experimental setup. I mean, we only had one set of data, and we didn't perform it ourselves. I mean, we're going off of some other class's data, so that could be potential errors. If you were to change the experiment, describe what you might do differently to give you more consistent or reproducible results. So, think about that. What could we have done differently? What would have happened if we had extended the experiment another week? What would have happened if we had included another treatment group that was kind of in between container size, that was a medium container with maybe a medium amount of food? How would that have changed the experimental results? What if we used some other species? So there's other things that we could have tried. So think of some other things that we could have done differently. So all of that goes in our discussion session. And then finally we get to our conclusion. So with our conclusion, we always want to think about what we learned about this whole experience. So, from this experiment, we didn't necessarily perform it ourselves, but what did we gain from this experiment? Okay, so what surprised us? And then what will we remember for the next time? Okay, so that goes in the conclusion. And then, just like the first lab report, we do need to have in-text citations throughout our lab report whenever we use information from a source. Okay, so that will be in APA format. And then we need to include a reference section at the end of our report. So that reference section must be in APA format as well. And our references, we need to have a minimum of 2 references. So definitely cite the workbook since we got the experiment from there. And then I suggest the textbook since we are learning about population growth and growth curves and survivorship curves and density dependent factors and things like that, all from the textbook. So you'll probably use the textbook to help you with that. And then if you want to get exemplary or the highest category for the reference section, you need to have 3 or more sources cited. So that you can choose any other source that is a credible source. So 3+ will give you that. Make sure to include those in-text citations throughout your report when you're using information obtained from a source. And then again, if you are having trouble figuring out APA format, there are a number of sources. You can just Google APA format and for sources or citations. I think if you just do APA citation format, you could get a number of sources to help you cite things. Okay, or you can just email me. I can send you a bunch of websites that'll help you. And then finally, proofreading is a good thing to do for any paper you ever submit to any class. So don't forget to proofread, spell check, look for any grammar mistakes. Everything should be in paragraph format, so don't use any bullet points or anything like that. Don't forget to indent your paragraphs. Typical 12-point font, normal font, so Times New Roman or something similar, double-spaced. And then don't forget your section headings so that I can clearly tell where your introduction ends and your methods section begins, since sometimes I can't tell for some of you if you don't include those. So that is it for the lab report. So once you are ready to submit, don't forget to grade your lab report against the rubric. Okay, so if we go into where we submit it, okay, so you're going to submit it under our topic 7 heading. Don't forget to actually grade it. Okay, so for some reason it went back to my teacher role, but if I go to my student role, it'll show you. So grade against the rubric. So that's why I kind of broke the introduction down into the different segments, because I have to grade it based on these different segments. Okay, so your hypothesis, it will be graded based on having both a regular hypothesis and the graphical hypothesis. Your method section, if you don't cite the workbook, it gets dropped down. Okay, so you have to cite the workbook to get the higher score. If you don't have the verbal and visual representations, so the graphs and tables named and numbered and referred to in the report, then I have to drop you down a grade. Okay, so you gotta number the graphs. You have to, refer to graph 1. In graph 1, you can see that, the line 1, the orange line representing the container with the large food and large, container size is doing blah, blah, blah. So you have to integrate that. Otherwise you get knocked down. So all of those things I have to grade. So make sure that you grade yourself against the rubric so you don't leave those important things out. Okay? And if you have any questions, feel free to email me or stop by my office hours because I'm here to help. I want you to get 30 out of 30 on these lab reports. All right, good luck everyone.